Welcome everybody to our Theoretical Statistical Ecology seminar of April 2023. Today we have the pleasure to introduce Laura Byrne. She's a PhD researcher at Trinity College Dublin, supervised by Professor Caroline Brophy, and she will be talking today about multivariate statistical models for biodiversity experiments. Thank you so much, Laura, for accepting our invitation, and the floor is yours.
Thank you for having me. It was a great introduction, so I think I can already go past this first slide. But in this talk today, I'll essentially be going into a few different topics. The first will be the biodiversity and ecosystem functioning relationship. The next will be diversity interactions models. Then we'll be going over some R tools for these models. And finally, we'll be looking at modeling some achieved proportions, with hopefully some time for questions at the end. To begin, the biodiversity and ecosystem functioning relationship is essentially the cornerstone of all of the research that I and many people are doing these days. It is defined as the way in which the species present in a system drive the quantity and quality of the goods and services provided. So some examples on the left there would be the different types of trees influencing the different gases in the air, or some plants inside of a field changing the amount of yield that field would provide. Multifunctionality refers to the provision of simultaneous functions by an ecosystem. So if we take that bottom example on the left, not only would these species be influencing the yield that comes from a field, they could also be influencing something such as the soil quality. And that would, of course, also be of interest to study, since we know that these ecosystem functions may have some sort of relationship between them. So quality probably affects the abundance of yield that you might get from a system. Why is the BEF relationship important to study? Well, there's two main factions that look at the BEF relationship, and that is agriculture and natural ecosystem specialists. In agriculture, the aim is to generally maximize the yield that a system provides. So for example, in Ireland, we have a lot of grazing land, which would want to maximize the food available to livestock. This goes hand in hand with a desire to minimize the presence of weeds and pests. This land will also likely continue to be used through future years, so we'll also have a great interest in the legacy effects that these different species may incur. In natural ecosystems, we may want to track changes in species composition to hopefully prevent these monocultures from forming by the competition between different species, weeding each other out. And that would also probably have an impact on wildlife, such as, ground cover, allowing for small mammals and insects to live in the ecosystem. How is it measured? So the BEF relationship is generally structured as the species diversity plus some structures, such as maybe walls or different divides like that, which lead to an ecosystem function response. Diversity can be defined in numerous different ways, the most popular of which is richness, referring to the number of unique species present in a system. So on the left, we have 4 different species. That's simply a richness of 4. However, it has a glaring weakness. That is, when the species are present in different levels, richness does not pick that up. In both of these situations on the screen at the moment, despite the fact that obviously the species are shown to be structured very differently, richness is still equals to 4 in both. Another popular method is the use of the evenness metric, which is the relative presence of each species. So it is maximized when all species are equally represented and then tends towards 0 as we approach a monoculture. This adds an extra layer to the diversity relationship. Diversity interactions models has its own definition of diversity, and that is the use of the initial abundance or relative abundance or proportions of the species in the system. So it is a regression-based approach, which can be fit like any other linear regression model using maximum likelihood estimation or least squares, for example. And it has the ecosystem function as a response on the left, followed by fixed effects of the species IDs, which are those proportions that I mentioned, plus any proportion-based species interactions or any extra treatments. Algebraically, this can be expressed as such, where M represents the different experimental units, or in our case, usually plots that any of these species can be planted in. Our first group of terms there is our species IDs, with coefficients beta I being each of the different species present. And in this way, the proportion PI is the initial proportion or relative abundance of species I, with coefficient beta representing how its presence affects the ecosystem function. Next, we have an example of species interactions, which in this case is the full pairwise interactions, where each set of two species are multiplied together and have their own coefficient delta ij for the species I and j, although there are many different options for species interactions. Next, we have a set alpha and A, which can just be any sort of treatments or block effects that may be included depending on the design. We then, of course, have our error term, which has a distribution normal with mean 0 and variance sigma squared. An expansion to this model could also be the inclusion of the nonlinear parameter theta, which allows deviation from the otherwise strict assumption that the species proportions must be directly proportional to the product of the species, or the interactions, would be directly proportional to the product of the species proportions. As you can see, it is represented as a power of that product. And it is quadratic in nature, so it would be bivariate products. There are other options, like I said, for these species interaction structures. And to illustrate them, I just want to give an example of some popular species used in Ireland, which would be perennial ryegrass, PG, Italian ryegrass, IG, red clover, RC, and white clover, WC. To make these names a little bit easier, I've just labeled them species 1 through 4. To begin, we have that full pairwise model, which was described on the previous slide, where each pair of species interacts in a unique way. So each and every one has its own delta value. Although this is an ideal model to fit when you have a small number of species, as the number of different species in your experiment increases, you're going to have a bit of an explosion of parameters, since each and every pair will have its own coefficient. So there are ways to simplify this interaction structure, such as using a functional group model, where we assume that each pair of functional groups, in this case, we can group our grasses and our legumes together, and each pair within those groups, so that would be the red and white clover or the perennial and Italian ryegrasses, and we assume that they each interact uniquely. So algebraically, we can express this as such, where G represents our set of grasses, species 1 and 2, and L represents our species of legumes, species 3 and 4. We can go even simpler than this, however, where we assume that all species interact the same, and in this case, we can allow this to depend on their average level of presence to get the AV, or the average interaction model. So that would mean that there is only a single term that represents all of these species interactions. Of course, there's also the option that no interactions occur between species, in which case we get the ID diversity interactions model. A further expansion to the DI modeling framework is the use of multivariate or repeated measures data in the models. This involves adding a few subscripts to the model that we've seen before, which include the addition of K for each of our ecosystem functions and T for each of our time points that may exist. Otherwise, it is structured exactly the same on the fixed effects side. So that is our response being in possibly biomass or yield and the soil quality that were mentioned before, where K would be each of those, and time points could be year 1 and year 2, meaning that our beta I coefficient is now the ID effect of species I on function K at time point T. Another interesting aspect of this modeling structure is actually in our error term, which is now a multivariate normal distribution with mean 0 and variance sigma star. Sigma star is a block diagonal matrix whose main diagonal is populated with a matrix sigma of size kt by kt, remembering that k is our ecosystem function number and t is our time points. And this represents the relationship between each pair of ecosystem functions at each time point. There are also some structures that we can implement on this sigma value. And in this case, if we had 2 ecosystem functions in a multivariate study, we could assume that they have an unstructured or generalized structure where we allow each relationship to be estimated independently. So that is each of those lowercase sigma values. And in a study with repeated measures with, for example, 4 time points, we could implement something called an autoregressive model of order 1, or an AR1 structure between the times, assuming that the correlation between each time point gets weaker by a factor of rho as the distance between the points increases uniformly. In the case that both multiple ecosystem functions and time points are present in the data, the sigma value can then be constructed by taking the Kronecker product of each of these matrices. That is, multiplying each element of the first matrix by the entirety of the second matrix, resulting in a matrix of a desired size of kt by kt. But it's all well and good to know that these models exist, but using them is the actually important thing. And to make this process as easy as possible, our tools have and are being developed for DI models. The first of which is a package that is currently available on CRAN called DImodels, which can automatically fit this modeling framework for univariate analyses at a single time point. It can be used with either the user's own data or any of the 11 included example datasets, which are a mix of both real-world and simulated datasets. There are 2 different fitting methods available. Auto-DI, which is intended for an audience with little to no modeling experience and will fit a range of DI models for a user based on their data input. It then performs model selection and outputs the suggested best model for the user to then explore. The DI model, or the DI function, will fit a DI model with specified interaction structures along with any additional fixed effects, and gives the user the option to have the method estimate a theta value for them or use a value supplied. It outputs the desired model as a GLM object, which is estimated using iteratively reweighted least squares. The package then also contains methods for further analysis, which you can see in the purple block here, such as predict, AIC, BIC, log likelihood, contrasts, or theta confidence interval, or theta CI. My work actually involves the development of an add-on package, DImodelsMulti, which enables the fitting of multivariate and or repeated measures DI models. Again, this can be either using a user's own data or any of the 5 included example data sets in the package. So a user can then use the main function in the package, DImodelsMulti, to simultaneously fit a range of these DI models in a similar fashion to the DImodels package, allowing the user to specify a single value for theta to be used on all of the models fit, or to allow for estimation of a different value of theta for each of the model types requested. The function outputs an object of custom class DImodelsMulti, which contains a list of the GLS objects representing each of the models. This object can then be used in some follow-up methods, just like the previous package, such as predict, AIC, print summary, and theta CI, or theta compare. Sorry, I forgot to change that value. That's supposed to be DI compare. But to illustrate the use of the package, I will use a dataset supplied by Dr. John Finn and Dr. Gillian Grange of an experiment of a grassland in County Wexford. This dataset contains 6 unique species, which could be grouped into 3 functional groups, which I did here for ease of illustration, and that would be the grasses, legumes, and herbs. It contained 43 plots of 6 monocultures and 13 unique mixes of either 2 to 6 species. There were 2 ecosystem functions available to be recorded, and that is the yield of the sown species and the yield of the weeds or unwanted invading species. It also contains 2 years worth of data, which I have broken down into just 2 time points of year 1 and year 2, but there are several harvests contained within each year. Results from previous univariate studies showed that functional group richness and changes in sown species proportions have a strong effect on the produced total biomass. An overview of what the data looks like would be like this, where we have a column for the plot, which is our experimental units. We have a year column for our time points. We have our 3 sown proportions from 0 to 1 of our grasses, legumes, and herbs, along with the yield of each of the species separated, and the yield of the sown species altogether, and the yield of the weeds. The input for the model would look something like this, where we include our species proportion columns, our 2 different ecosystem function columns, Y. We specify the different types of structures we would like to implement between our ecosystem functions and our time points. In this case, for ecofunc, I chose to have it be unstructured to allow each of the relationships between these ecosystem functions to be estimated without constraint. And for the time, I implemented an AR1 structure. We then specify our experimental units as plot and choose what type of interaction structures we would like to model. In this case, I simply input ID for the ID model, full for the full pairwise model, and AV for the average interaction model. We then simply supply our data set of grassland. Now to actually follow the journey of this input through the package. Beginning with our data input of grassland, we do some error checks, make sure everything is looking in tip-top shape, everything is the correct data type that we want it to be, with our proportions being integer values that should add up to 1, but that is not a harsh throwing an error kind of stop, because there are some experimental setups where you may want your species to not actually fully add up to 1 and have a separate fixed effect term allowing the actual sum to 1 constraint to be fulfilled. But we then also reformat the data to be stacked. So in the case of this data set, we have our different yields types, our yield sown and our yield weeds in different columns. This is going to change it to have a column indicating the type of ecosystem function and a column indicating the value of that ecosystem function. Next, we want to add in the different DI model interaction columns. And in this case, it would be our full pairwise and our average term, but it could be any of the different columns available, such as evenness or the additive interactions. It simply slots those into the data set for you. Next, we would construct our fixed effect formulae and our correlation structures, which may look something like yield value interacts with our yield type and our year, summed to our zone G plus zone L plus zone H plus our different interaction columns, which we will swap out depending on which model we're currently doing. This formatting of the formulae essentially allows for a different coefficient for each of our species IDs and interactions, depending on the ecosystem function and the time point. For our correlation structure, we will then make a core struct object following instructions from the NLME package. We then pass all of this information into the function GLS from the NLME package, which utilizes generalized least squares as an estimation method. It then also allows you to specify whether you want to use ML or REML, which the package also allows you to specify in order to pass that information through to GLS. We then simply add all of the models that we fit to a list and convert that into a DImodelsMulti object, returning that object to the user, who can then use any of those follow-on methods to further investigate. An example of the output that this would get would be some notes on what it is that you input, because some of these estimations can take a bit of time and maybe you forgot or you did it a few days ago. And this is just to make sure you're aware that in this case, REML was used. So it would probably be best to not try any fixed effects comparisons between the models that you achieved. And then to let you know what correlation structure was used, along with what type of model was fit. You would then also get a fixed effects coefficients table, including all of the fixed effects, and along with the residual standard deviation and a significance indicator. You then may also get a variance covariance matrix, along with a bunch of other information that there's just too much to fit into a single slide. Onto our achieved proportions data. So in this case, we could look at a univariate DI model, which has a popular ecosystem function of yield, especially in the agronomic communities. And this may be directly relative to our individual proportions when you break it down. So inside of this yield, there are several different species, be those the ones that we sowed, our PI values, along with any weeds that may have invaded their way into the space. And yeah, this may be directly relative to our starting proportions P, and we could just all go home and know that they had the same relationship, they took up the same amount of space throughout each time point. But it's kind of an ideal world situation, and it is more likely that these species presence values or the species composition will change over time. This could be due to the selection effect, having some competition between species where some just establish faster than others and then take root and do not allow the others to thrive, or anything of that sort. It could also be environmental. Perhaps the weather was too dry for some species, too wet for others, and others just managed to take over the space. Anything like that could change our species composition. And basically, we may want to track that just because over long periods of time, it is possible that our initial proportions will no longer be a strong enough indicator of the diversity that is now driving the ecosystem function we are studying. I mean, some experiments have been going on for over 20 years, and it may be a little bit facetious to assume that our initial proportions will still be the strongest indicator, as opposed to maybe the proportions of the species from 10 years after. Anyway, the proposed model for actually fitting this breakdown of the species proportions within yield can be built up from the basic DI model. Here I'm using the average interaction DI model as an example. Our first step to this expansion would be to add subscripts C and M for our species that would be inside of the yield as C, and our experimental units M to keep track on the relationships within plots. This also allows our epsilon to expand to allow for that relationship that we saw with the multivariate repeated measures model. We can then add a further subscript T for each time point, one of the most important factors of this study. We can then also add any extra info, such as the previous total of the yield from any previous year. We could add what the proportion of previous year's total was weeds, or anything that may have changed between the time points that we think could affect the species composition. In order to illustrate this, I'll be using the same data set as previously, but with a bit more fine-tuning for the sake of illustration. We still have our 6 species from this grassland in County Wexford that we break down into 3 different functional groups of grasses, legumes, and herbs, with our same 43 plots with 6 monocultures and 13 unique mixes. We, instead of having 2 different ecosystem functions, have a single ecosystem function of our dry matter yield, which we can break down into our 4 components, which is our grass yield, our legume yield, our herb yield, and our weed yield. And then we have 1 year of data, just to make plotting the results a little bit easier on myself. I have a colleague who is working with visualizations for DI models, but sadly, I am not the expert in visualizations yet. So the same results apply from this data set, where previously we found that functional group richness and changes in zone species proportions have a strong effect on the produced total biomass. This modeling framework will essentially allow us to dive a little bit deeper into what goes on past just the changes on the total biomass. What is that biomass made up of, and is it desirable? What is inside of that biomass? So for again, an overview of what the data set looks like, we just have our plot, which is our experimental units, our year, which in this case we only have 1, and our sown species, and the yield for each species and weeds. The results of an analysis using this model, using this modeling structure, resulted in the full pairwise model being best, likely because we only have 3 different species to interact, allowing us to have not an explosion of parameters in the model. I also added a constraint to the model, which kept the resulting YCM value as 0 if the species was not sown. That was simply adding in a factor which interacted with each fixed effect and toggled to 0 if the species was not sown, and 1 if the species was sown. Essentially, we found that herbs being included in any way with other species present developed interactions which reduced the yield of weed inside of our total biomass. And the presence of legumes when interacting with grasses improved the grasses' yield. This is likely due to the fact that legumes have this natural ability that is quite unique to them, allowing them to fix nitrogen from the atmosphere and make it available to the soil and all other species within it. So we all know that grass loves nitrogen based on the nitrogen fertilizer prices just skyrocketing recently, and farmers trying to find any solution to not have to buy quite so much of this treatment. Well, apparently legumes can allow your grasses to thrive in a similar fashion. So to actually plot this out, I took a selection of communities that may be of interest and predicted for them from the resulting model and fit them into this plot. So we have each of the monocultures, the first being grass, the second being a legume monoculture, and the third being a herb. And what we can notice here is that if we were only looking at the total biomass without the breakdown, we may assume that the best performing monoculture is the legumes, because I mean, that bar goes nice and high above the other two. But with the breakdown, we can notice that a large portion of that yield is actually weeds, which may be unwanted, because if we're thinking of grazing land for cows, it could be some less nutritious plants. It may not be our Irish famous grass or any intentionally planted legumes. It's just this unwanted species. And so our actual proportion of legumes that was inside of that yield is lower than our grass or our herb monocultures. But when we go to our 50-50 communities of mixing a grass and legume evenly, we can see that the total yield increases greatly. We actually call this over-yielding because it achieved better than any of the individual monocultures. And we can also see that some of the interactions between the two did actually reduce the weed presence. That yellow bar is significantly smaller. We see an even better reduction when it comes to our fourth bar, which is a 50-50 community with grass and herbs equally present. And we can see our grasses aren't performing quite as well as they were with the legumes, likely due to not having that extra nitrogen, but our weeds were greatly reduced compared to the grasses monoculture alone, which is generally the case in Ireland, just having lots of grass everywhere and nothing else. But when we reach a richness level of 3 and have all 3 species equally present at 33% each, we can see that we get a very similar proportion or level of yield for our 3 desired species as our grass and legume mix, but with a further reduced amount of weeds present. So our 50-50 grass legume has the same desired yield as our 33-33-33 grass, legume, and herb, which would likely mean that we still have a bit of space to grow in that field. Maybe we could plant something else to take in that space. We have less bare ground or something of the sort. For future work with these achieved proportions, we could look at the difference between the sum of the estimated breakdown and the univariately predicted estimated total, and see if there's any way to include some further restrictions to increase this model's accuracy. Because in the end, we do want our individual species yields to add to our total if we're having a perfectly accurate model in by comparison. Obviously, nothing will be perfectly accurate, but we want it to get close at least. So in summary, we talked about the BEF relationship and its importance to study. We also know that there are many different ways of defining diversity, and one of the things that are looking at this definition of diversity is diversity interactions models, which is regression-based and utilizes the initial proportions of species as the main driver for change in ecosystem functions. We also know that there are R tools available for DImodels, which includes the currently available on CRAN DImodels package and the soon to be available on CRAN DImodelsMulti package. The first of which allows for univariate fitting at a single time point of these models. The second of which will allow for multivariate and repeated measures models to also be included in the mix. We then know that achieved proportions would refer to the specification of the breakdown of yield over time and its prediction, along with the importance of noting that maybe looking at an ecosystem function such as yield as a whole isn't the most ideal when you don't know what that yield is comprised of.